Welcome back, friends. In this session we are going to continue with our coordination session, and today we are going to discuss about the different experiments done during the discovery of auxins. So as we said in the previous session, that auxins are the first scientist to investigate about auxin was Charles Darwin, and he did different experiments about auxin, and in that way they knew that the growth of coleoptile is influenced by different hormones and is influenced by the chemical substances produced by cells, and probably is there from the coleoptile tip. So we have this table, and this table is coming from the Understanding Biology book. So this table shows different experiments done by scientists, and by this table you can know how they became knowing about the auxin. So the first four experiments they were done by Charles Darwin in 1880. So if you look at this table, we have the researcher, we have the method, we have the results, and then we have the explanation why they got those results. So first we have Charles Darwin in 1880. How he did: first he took the coleoptile tip, which is straight, and then he illuminated with unilateral light. So unilateral, that means light coming from one direction. Now probably this side was dark, and then unilateral light was illuminated on the coleoptile tip. What happened? The coleoptile bent towards the light, and the explanation of this phenomena is that the coleoptile is positively phototropic. Bending occur behind the tip in the region of cell elongation. So probably according to this experiment is that the distribution of auxin from the tip to downwards, it tend to escape the side of light. Kwa mfano, kwa sababu walikuwa wameilluminate upande wa kushoto ulikuwa hauna auxin. Is auxin zinazokuwa produced from the coleoptile tip zina distribute downward, lakini zinakuwa zinakimbia mwanga. Kwa maana yake zikidistribute hapa zitadistribute kwenda right hand side, na zikidistribute kwenda right, hapa chini ndo kuna region of cell elongation. Kwa maana yake auxin zikija kwenye region of cell elongation, zikistimulate cell elongation, that means this right hand side will glow or will elongate more as compared to the left hand side, and that more elongation of the right hand side cells will lead to the bending of this coleoptile tip to the left or towards the light, and we explain it as positive phototropic effect of auxin. But this positive phototropism is because of the property of auxin ambayo inakuwa they are distributed away from light. Now the second experiment wakataka kujua kwamba now tumeshaona kwamba in the presence of coleoptile tip, and by illuminating it with unilateral light, it bent to the left, and then wakathink more kwamba where does this chemical substance comes from. So they cut out the coleoptile tip, and then walivyoiondoa coleoptile tip, after removing the coleoptile tip, waka illuminate again the coleoptile. Kwa walivyo illuminate coleoptile wakaona kwamba no response. The results of the experiment, no response. Why there is no response? Because the region of cell elongation still is present, but why there is no response? It is because the chemical substance which influenced the cell elongation is probably produced at the coleoptile, which is already removed. Now the explanation for this results is that the tip must either perceive the stimulus or produce the message, or both, as it is removal prevent any response. Coleoptile tip hapo inaweza ikawa either ina produce hormones au either inaweza ikawa ina perceive message, manake na act kama receptor ina perceive the message of light illumination, and then it causes the distribution of auxin. However, according to the further studies, mpaka baadaye walivyokuja kufanya conclusion of the auxin, ni kwamba auxin they are produced at the tip and then they are distributed below the plant. Kwa maana yake ni kwamba removing the coleoptile tip, tulikuwa hatuna tena auxin ambazo zinaweza kusababisha cell elongation. So coleoptile tip sio kwamba ina perceive the message, but it tend to produce the Auxins. And the third experiment done by Darwin, alifunika ile coleoptile, alifunika coleoptile, akafunika na kitu ambacho kitazuia mwanga usiguse coleoptile. Now kwenye experiment alifanya light proof cover is placed over intact tip of coleoptile, hafu waka illuminate again with light. I told you kwamba Auxin distribution ina depend kwenye light. Tukiwa hatuna light, maana yake Auxin zita distribute kwenda downward, lakini hazita distribute unilateral. Kwa distribution yake itakuwa kwenda downward, lakini distribution ya kwenye left hand side na kwenye right hand side itakuwa ni sawa. Tukiwa tuna right, distribution ita distribute upande ambao hauna mwanga. Kwa hiyo hapa, kwa sababu tulikuwa hatuna light, kwa sababu gani tulikuwa hatuna light? Kwa sababu the stimulus of light is perceived by the coleoptile. So ukimask ile coleoptile unakuwa mask uwezo wa plant kuweza kudetect stimulus. So we end up with no results. Why? Explanation for these results is that the stimulus must be perceived by the tip. And then the last experiment done by Darwin, walifukia sehemu ya ule mmea, kwa wakaweka ule mmea kwenye fine black sand, wakafukia afu wakaiacha ile coleoptile tip iwe open, then waka illuminate again with unilateral light, ukitaka kuona kama mmea utabend au utabend. But after that experiment wakaona kwamba mmea ulibend kama kawaida, yani kufukia kipande cha chini mmea haukuwa affected chochote. That means the stimulus is perceived by the tip rather than the region behind it. Ndio maana tulivyofukia region behind the tip, no any effect ambayo ilikuwa imetokea. So that means the stimulus is perceived by the tip. The distribution of Auxin to unilateral side took place as usual, and then the results was phototropism. Now we have the Boysen-Jensen in 1913. He did other experiments again demonstrating the properties of Auxins in plants. Now the Boysen-Jensen, he used the thin impermeable barrier of mica in his first experiment. Now in his first experiment, he placed the mica on the side of illumination. He placed the mica on the side of illumination, or the illuminated side of the plant. And as I told you that Auxin, they tend to distribute away from the side of illumination. So mica is impermeable, does not allow any water soluble molecule to pass through it. Now if it is impermeable, that means if it is placed on the side of illumination, it will not prevent, or it will not reduce the distribution of Auxin to the region of cell elongation. Now after illuminating the plant, then the Auxin, they distribute to the right hand side of the plant. And the results of the experiment is that the plant bent as usual, because the distribution of Auxin is away from the side of illumination. Now movement of hormone downward on the shaded side, as you can see in this diagram. And the explanation for this experiment is that mica on the illuminated side of the coleoptile allow the hormone to pass down the dark side only where it increases the growth, on the dark side only where it increases the growth rate and cause bending of the plant as usual. Kwa maana yake ni kwamba mica hii hapa, kwa sababu ni impermeable, lakini imeweka upande wa illumination, kwa Auxin zina distribute kwenda kwenye shaded side. Now look on the second experiment, and then you will see the difference between the first and the second experiment, and what is the importance of inserting mica on the illuminated side. Now aka insert mica kwenye shaded side. Experiment ni ile ile, kilichotofautiana ni side of insertion of mica. Alivyo insert mica on the shaded side, then aka illuminate. So movement of hormone down shaded side is prevented by mica, because mica is impermeable. Alivyo prevent, kuna no response, no phototropism. Tuna experiment nini kutoka hapo? Manake mica is distributed on the shaded side. As mica conduct electricity, it would not prevent electrical message passing down from the tip. So it will however prevent chemical ones, because mica can allow electric current to pass through it but cannot allow chemicals because it is impermeable. So if there is no response, the message must be chemical and must pass down the shaded side. So from this experiment we are concluding two things: that the message which is produced here, it is chemical, and the second thing is that it must pass downward the shaded side. And then we have the third experiment in which he used the what we call it, the gelatin block, and in this experiment we have the tip removed, and then he put the gelatin block, but the gelatin block is permeable to hormone. So because it is permeable, it allow the hormone to pass through it on the shaded side, and the phototropism took place as usual. So the tip removed, gelatin block inserted, and the tip replaced, but the gelatin block is permeable. Now movement of hormone down shaded side occur as usual, as gelatin block allow chemical to pass through it but not electrical messages. The bending which occur must be due to a chemical passing from the tip. So that means the gelatin block comes to prove the point that the message is chemical in nature and not electrical. Now when we are going to finish the discussion of this experiment, let's discuss about the synthesis and distribution of auxin. Auxin, as we have seen in the experiment, auxin are made continuously in the shoot, apex and young leaves. Shoot, apex and young leaves. Movement away from the tip is described as basipetal. Basipetal, that means it is from the apex to the base of the organ, from the apex to the base of the organs. So and the polar in one direction only. So movement of auxin, it is from the shoot, apex and young leaves to the other parts of the plant. Na kama ulivyoona kwamba tumeelezea movement is basipetal, that means from apex to the base of the organ, and it is polar, that means in one direction. So it moves apparently by diffusion from cell to cell, and is eventually inactivated and degraded by enzymes. That means kama ikishafanya kazi imemaliza, it is degraded by enzymes. Long distance transport can occur via vascular system, mainly phloem, from shoot to roots. Erythroauxin is probably made in roots. Erythroauxin, lakini higher concentration of auxin, it is made in shoot, apex and young leaves. It is very important to know about the synthesis and distribution of these phytohormones, because some of teachers can ask you to explain about chemical structure or the discovery of these hormones and their synthesis and distribution and even their laws. Now in this session I am ending up here. In the next session we will finish about the auxins by discussing the laws of auxins and their commercial application. Thank you everybody, and don't forget to subscribe and to share this video to your fellow students. Thank you. I wish you nice studies.